And we're off. This is the serotonin chapter. Uh, Serotonin was arguably my first neurotransmitter love before I moved over to opioids. And I'd like to draw your attention to this picture. Later, we'll see other images that deal with this part of the midbrain. Um, this is the dorsal raphe nucleus. It's dorsal, suggesting there's another one. It's not ventral, it's medial. It's down below. Notice it seems like a crease or a seam down the middle of the picture. Raphe means seam, and there is no left and right, which I know I've mentioned in an earlier lecture. And here you can see individual serotonergic cell bodies dotted around and also fibers. So this is a very nice picture, and that is classic. And this, we owe Dahlstrom and Fuchse thanks again, because they are the ones who first visualized and named these using letters as they did for the catecholamines, but for serotonin they called them Bs. And the book focuses pretty much on the dorsal and median raphe. There will be some mention of raphe magnus, a favorite of mine also, and you might be able to guess why it's called Magnus. It's big, and it's these raphe here are important for projecting serotonin rostrally in the brain toward the forebrain. Magnus is very important for projecting serotonin into the spinal cord. And this is a pretty dramatic and eventually fatal story. And someone I care about was just temporarily diagnosed with this not too long ago. But it's serotonin syndrome, and it can be, quite the adventure. Um, this woman got it because she was overdosing on drugs relating to increasing serotonin activity, and eventually died. This is a problem for those taking serotonin agonists therapeutically, but it's not huge. And I feel a little uncomfortable that they've opened the chapter with, death from serotonin. Now, we just came out of the catecholamine chapter, which was the last chapter in block 1. And this should look kind of familiar because that's the way the last chapter, chapter 5, dealt with the catecholamines. There's a discussion of synthesis on the left, and there's structures on the right, and you don't have to know the structures, but you should know the names of the enzymes for sure, and the steps of serotonin synthesis. Just take a look at the structures, and you can see what's being modified with each step. Um, can you remember the amino acid that's important for synthesizing catecholamines? It also starts with T, as is the case here. And there it's tyrosine, and here it's tryptophan. L-tryptophan, that's another story that we won't get into right now. But tryptophan, and like tyrosine, it's an essential amino acid, and it's called essential because you don't synthesize it. You have to get it from your diet. Um, and I just went through this, and I didn't see this in the chapter, but normal corn has no tryptophan. And if you get a lot of your caloric intake from corn, you may be low on tryptophan, and that could affect serotonin levels. In fact, early on in research on animals back then, they would have been rats, to produce a decrease in serotonin. You would feed them specially formulated rat chow that was tryptophan free. And it took days, of course, but over the course of days, brain serotonin levels would go down quite a bit. In the last chapter, there was a discussion of rate limiting steps, which are important, which is important for uh, you know, virtually any neurotransmitter. And here's a nice quote: just as the initial step in the synthesis of dopamine, that is tyrosine to dopa, is the rate limiting step. The conversion of tryptophan to 5-HTP is the rate limiting step. And the first research drug I ever used attacked that step. And I think I've already referred to it, and we'll see it again in this chapter. I was struck. I hadn't really thought about it that much. I was struck by the number of parallels that there are between the catecholamines and serotonin in terms of their life stories. So if you mastered the concepts of the catecholamines in the last chapter, serotonin should be much easier to deal with. And of course, they are all monoamines. Only the catecholamines are catecholamines, and serotonin is a monoamine that's not a catecholamine, but their life stories are quite similar. And going back and forth between those two is worth the trip, I believe. There's our friend, the blood-brain barrier, which I asked about in a quiz a bit ago. You have to be able to get things into the brain, and tryptophan will not just go in there. It has to be transported. And that transport process gives us some interesting twists that we can use for research on serotonin. And we do this sometimes to undergraduates. We give these little cocktails to undergraduates that make it often that tryptophan is not able to enter the brain for a little bit, and they do affect behavior. And there's a nice discussion there from earlier on. Fasted rats, meal low in protein but high in carbs, increase in brain tryptophan and consequently serotonin levels. Nice story. Oh, look, the blood-brain barrier again. And remember, this is caused by the epithelial cells of the capillaries having tight junctions, and everything has to go across the outer and inner bilipid membranes. And here there's got to be transport mechanisms. So there's dietary tryptophan, which makes L-tryptophan accessible. But other things like large neutral amino acids can be competing for the same transport mechanism. And if you flood the transport mechanism with large neutral amino acids, they win. And the tryptophan doesn't get in, and consequently, the synthesis tryptophan to 5-HTP to 5-HT is reduced. This discussion of insulin is very interesting. And more on dietary intake and stuff. What I used to believe that it was really unlikely that fairly trivial changes in diet could substantially affect neurotransmitter levels in the brain. That just doesn't seem smart on the brain's part. You can't run out of things. But these things do work, but they are pretty contrived. Normal food intake is not going to cause these effects, but it's definitely worth reading. And we, as I said, we do sometimes use it in research on undergrads. And there's my first drug, da-da-dum, PCPA, parachlorophenylalanine, selectively blocks serotonin synthesis by irreversibly. Now, many drugs, when they interact with receptors or membrane elements or cytoplasmic elements, they bind reversibly. That is, they bind and they fall off, which is the way most drugs work. They hit the receptor and they fall off, but not PCPA. It irreversibly inhibits TPH, and as it says, 1 or 2 high doses, and I think I only did one, can reduce serotonin levels in rats by 80 to 90% for as long as 2 weeks. And I remember possibly my first live talk at a meeting. I had given PCPA intraperitoneally, and you might look back at that in another chapter in terms of how to administer drugs common with rats, injected into the peritoneal cavity. The rats did not look good after that. Serotonin, as we'll see, doesn't simply exist in the central nervous system. It's in a lot of places. And that's a very aggressive thing to do. I would not recommend it these days, and I don't think it's used very much anymore. So there's more on cocktails and amino acids and tryptophan. And here's tryptophan loading. In healthy subjects, the effects of elevating tryptophan on mood and cognition seem to depend on the magnitude of the increase of tryptophan to LNAA ratio. It's reinforcing my earlier bias. Even when you do this very systematically, the effects are gentle, but they are predictable. Modest has little effect, intermediate has some enhancement, and then high, anger, tension, depression. And so it's complicated and not totally understandable, but certainly true. More cocktails. And yeah, we're not going to get to the chapter on depression unless something dramatic happens in this course, because that's chapter 18. But the good news is that you're getting information on depression and schizophrenia and anxiety throughout the textbook. So I'm sure that some of you have interest in this because you or someone you know have depression or anxiety, or you just want to understand psychopharmacology. So there's a nice discussion there, and you see that serotonin is important, as we know, but norepinephrine is now also thought to be important. Have no fear, I have a battery charger, and we are now on the battery charger. Similar processes regulate storage, release, and inactivation of serotonin and the catecholamines. Again, if you mastered the last chapter, this should not be a very big deal. So there's tryptophan hydroxylase catalyzing the reaction to 5-HTP, AADC catalyzing the reaction to 5-HT, and then we store it. And I told you the story probably in the last lecture about my friend who left his research data in the hall and didn't store it in his room when he went to graduate school in Binghamton, and maintenance came along and threw it away. So storing the serotonin, moving it into the vesicles is very important. And you can see that there are special mechanisms for that. And don't forget that those vesicles are a bit like little cells. They have bilipid membranes. and they have to move over and live near the presynaptic release point where they fuse but do not leave the cell, forming omega bodies because it looks like omega. And there are the postsynaptic receptor sites. So, purple is the presynaptic cell, blue is the postsynaptic cell. Most of these receptor sites are metabotropic, which is graphically what they're representing there. There are some ionotropic, which you should know. There's a 5-HT transporter, SERT. SERT is very important, and we can knock that out, and that has dramatic effects on animals. If you block that receptor, you can't get serotonin easily out of your synaptic cleft. It can diffuse away, of course, but the SERT transporter mechanism is much quicker. So drugs, there's SSRIs, which have been alluded to before, and they show up heavily in this chapter. They slow down reuptake, therefore lengthening the impact of serotonin on the post. Oh my God, on the postsynaptic receptors. Let me say this about that. You take morphine, an opiate, although the book would call it an opioid, and morphine would directly go in here and stimulate postsynaptic receptor sites. And independent of what the presynaptic cell wanted to do today, it's really cut out of the picture. The drug, the ligand, that hits these receptors, is directly going into those receptors. They are responding to the drug regardless of what the presynaptic terminal wants to do. It's hard to imagine people being very normal if you flood postsynaptic receptor sites with drugs all the time. I mean, we do it. It can work, but you don't hear very well if you just turn up white noise and blast it into your head. It works, but it's not very normal. It's not like the normal functioning of the nervous system. Now, if you block reuptake, which is going to have an agonistic action because the serotonin stays longer in the presynaptic cleft, and therefore the molecules get to hit the postsynaptic receptor sites multiple times, that serotonin is only going to hit the postsynaptic receptor sites multiple times if that purple cell decides to release it. You are speaking with your own voice. So I liken this to using a megaphone. So you want to say something to someone who's far away, you get a megaphone, you talk into the megaphone, your voice is amplified, and your message can sometimes quite clearly be transmitted across a fair distance. So it makes sense to me that drugs like SSRIs, which block reuptake, or at least inhibit reuptake, they can have therapeutic effects because your serotonin neurotransmitter systems are doing what they normally do. You're just giving them a louder voice. That to me seems much more likely to be therapeutic than flooding directly postsynaptic receptor sites with serotonin. You're not going to get normal behavior often with that. Now, the 5-HT autoreceptor, we've seen these before, autoreceptors. What's their function? And I don't think I asked about it yet, but I might. I discussed old people who have failing hearing. And if the 5-HT auto self receptor is supposed to listen to serotonin, and you take a drug that disrupts that receptor, blocks it, you can't hear your own serotonin. So you might release more. Or if you had a drug that stimulated that autoreceptor, that tells it, oh my God, you're releasing so much serotonin, then that presynaptic terminal is not going to release as much. So autoreceptors are very important. By the way, figure 6.3, um, this is really frightening. The number of serotonin receptors that exist, it's truly amazing. When I left the field, we had no idea that there were this many. So there's all these types and subtypes, and they do things and they don't do things. And one of the questions at the end of the chapter is to list them all. That would be daunting with the book closed, but with the book open, it's not so bad. And there's PCPA again, fen-phen, which they used to have people take as an appetite suppressant. That didn't go very well. MDMA, ecstasy, lots of discussion of that in here. We'll see that in a bit. Inactivation of serotonin, again, many similarities to catecholamines. You have serotonin transporters, SERT, fluoxetine, Prozac. That's all, we sort of alluded to that when we were looking at the blue cell and the purple cell. Cocaine and MDMA interact with SERT, but they are not selective. They affect both, also the catecholamines. And here we go with our knockout mice or mutant mice. I'm not sure that they need to be knockouts. An astonishing array of behavioral and physiological abnormalities from chronic enhancement of serotonin activity. Now, if you don't have functional SERT, that's not just today. That's while you were in the womb. That's while you were growing up. The impact of this normal behavior can be profound, way more dramatic than temporarily interacting with SERT. So, if you or someone you love is taking an SSRI, don't get freaked out when you read about what happens with these mice that don't have SERT. Okay, now there's a big box, ecstasy, and here's some structures, and you can see that MDMA, MDA, MDE, amphetamine, and methamphetamine, there are some structural similarities, and there's a nice discussion here. When I went to UCLA, there was this part of the psych building that seemed odd. And word on the street was it was odd because your federal government paid for it. And they were doing secret research in there. Our military and the CIA were pretty interested in hallucinogenic drugs for a while. And they paid for a lot of research that was secret. And this discussion is of research that was withheld from public view for 20 years. Interesting. And by the way, that 20-year window upsets me because there's a 20-year window in understanding something that I did, as we'll see in a bit. So that's interesting. And look at that. I forgot to highlight that, but Merck, still a famous drug company, synthesized MDMA in 1912. Nice going, Merck. They have all these chemists. I imagine these underground warrens filled with chemists twisting and creating molecules. Maybe it's just me who thinks that. This is scary. A couple of years ago, MDMA was fairly popular. And I used to have a discussion board. And please, you guys are not using the discussion board. Please use the discussion board. And I have a discussion board up for every chapter. So when you get to the questions and I use the words in the question for the quiz, you should not at that point be expressing confusion over what the word means. If you have a question, post it. And our learning community can get to work on it and help you understand what it's about. Now, we saw the first picture in, I meant to, this is dark field again, darn. And I did not do the search on this. Let me quickly get you a dark field. Because I mentioned this before, and I think I did a fairly good job of describing the difference between light field and dark field, but perhaps not the best. Okay, this sort of tells the tale. So you've got your specimen, and you have special objectives that make it so that the light coming through the specimen, unless it has shiny spots on it, does not go up through the objective. It goes off to the side. Therefore, instead of having a light field, you would have a dark field. Because if you have a clean piece of glass and tissue that does not disrupt the passage of light, and by disrupt, I don't mean block, it has to reflect it a bit. What you see with your, I'm sorry, this is going down. So the light is going down, which is confusing for me, but not for you. But again, you have light that is not going directly through the specimen. And so your light source is up here, not your eye. The light comes down and it goes everywhere except where you're looking with your objective. But the light is going around it, forming a cone of light, because the light is, like you draw strings through a dot, and the strings all go off to the side. None of them go through the objective. But when there's something shiny in here, some of these cones of light that are missing the tissue, they will be reflected up, and you will be able to see them. That was not the best example. Here's another one. That's more better. So with dark field, you of course get a dark field, but where you have, in this case, serotonergic fibers, they have been marked with antibodies, and the marker that goes on the marker for the antibody has a crystalline structure. So when you try to shine light through it, that crystalline structure will, randomly ding some of those light rays, and some of them will go up through the objective and you can see them. So in this picture on the left, and this has to be, does it say this really should be, yes, visual cortex. And this is a primate. Notice that there's a lot of staining in the normal tissue up here. That's lamina 1, the first layer of the cerebral cortex. It's a layer we almost never talk about, but serotonin loves it. And look at that, man. Would you take a drug that can do that to a neurotransmitter system in the brain? Now, this is overkill, and the book tries to pull back on this. But boy, once I saw this, it really frightened me. This is not a drug to be taken lightly. It may have therapeutic effects, legitimate therapeutic effects, but it should not be taken for fun. I vote. So they have declared MDMA back in '85 as Schedule 1. Now, some drugs are Schedule 1 for racist reasons. And yeah, but this is based on science. And there may be some use for this. This is an aggressive use of the drug. But I used to run a psych conference, and a student came and presented. Actually, he missed his presentation and came years later when he had his PhD. And he had studied rats who had been given the equivalent of a weekend's worth of ecstasy. And he did not show this. He used Golgi stains, and we were looking at axon terminals, dendritic branches, and they looked like nothing I'd ever seen before. They looked like bunches of grapes. So I'm still pretty afraid of this. But if you have a debilitating disorder, I guess I would think about it. And a lot of talk. This is an important drug. And at the end, it says, nevertheless, it is intriguing to consider that we may have now come full circle with MDMA, originally discovered in a program to develop new therapeutic compounds for homeostasis. It was later found to be neurotoxic when used recreationally at high doses, and now considered potentially beneficial for patients with severe treatment-resistant PTSD when given at low controlled doses. The first student I worked with when I came to Scranton, he was a Vietnam vet, and he had PTSD, and I didn't even know what it was. When there was a thunderstorm in the area, he would go into part of the lab and go under the table and shake. And you guys largely have lived with us being at war during your entire life. PTSD is really real and really awful. And for that purpose, I would be willing to consider many things to help. Now there's there were knockout mice, CERT knockout mice. Look at all that stuff. Just let it wash over you. Hearts, intestines, hypoalgesia, thermal nerve injury, gut hypermotility. When I was you, we never thought about the gut. But now we know that lots of neurotransmitters that are used in the central nervous system are in the gut. Here's an interesting fact. I produce a lot of saliva. That's maybe too much information. But people who are clinically depressed don't produce much. So think about that. And look at all this stuff: stress, susceptibility, ACTH, epinephrine, seizures, REM sleep disorder, depression, somatosensory systems, underaggression, late onset obesity. Serotonin is not just in your brain. It does lots of things. You would like it to be in the normal range, so you can be in the normal range on all those things. 5-HIAA. That's a breakdown product. So serotonin is not a catecholamine. It's not affected by COMT, which you read about in the last chapter. However, its breakdown is catalyzed by MAOA to yield the metabolite 5-hydroxyindoleacetic acid, 5-HIAA. And you can measure that, as was discussed in the last chapter for catecholamines. You can measure that in CSF, and there's a really bad story about them measuring that in somebody who was hospitalized with suicidal ideation, and his 5-HIAA levels went nuts, And by the time they called him during his release, he had committed suicide. Although, getting things out of cerebrospinal fluid is not trivial. Okay, neuroanatomy. Now, the last part of this chapter is going to be a lot of drugs and stuff. And the book does as good a job as needs doing, and I really can't add very much to that. But up to this point, I've lived this life, and the neuroanatomy, I've lived that life. And the Swedish researchers who first mapped the catecholamines in the 60s, see Chapter 5, Dahlstrom and Fuchse. And here's the discussion, which I've already alluded to when we were looking at the raphe, dorsal raphe. Here they use B's instead of A's. And this is not immunohistochemistry, antibodies, tagging things. It's not the modern way to do this. It's paraformaldehyde gas that causes the monoamines to glow, and they do not glow brightly. And as you look at them with a fluorescent microscope, you use up the fluorescence. So you've got to move quickly, and the color differences can be quite subtle. So you have to be good to do this. And there's the raphe. And I've already mentioned that raphe means seam, and we've seen the dorsal raphe. And of greatest interest to neuropharmacologists or psychopharmacologists, which is what we're studying, are the dorsal raphe nucleus, and that was the poster child for this chapter, and the median raphe nucleus, which is also located in the midbrain. That's a rat, so we'll stick with that. And it's in people too, midbrain, just below. Together, these nuclei give rise to most of the serotonergic fibers of the forebrain. And we've seen the dark field images of those fibers. And we're about, to learn something about what we've already seen in the chapter. Okay. Right. They're not uniform. Of course, they go to certain places. Serotonergic fibers in the mouse neocortex. You would probably just call that the cortex. It's the cerebral cortex. And the hippocampus. And boy, the hippocampus is important. And we've seen some of that in relation to spatial memory and memory in general, navigation, spatial orientation, and memory. Note that in the cortex, the fibers are particularly dense in layer 1, which I've already mentioned, which is a layer that has very few nerve cells, but it has a lot of dendrites and synaptic connections. I almost never think about lamina 1, but it's just layered with, it gets a slew of serotonergic inputs. Very few fibers in the two major cell layers of the hippocampus, the pyramidal cell layer, and the granule cell layer. That's interesting. And pyramidal cells, that should ring a bell because they look like pyramids, and they have apical dendrites and basal dendrites, and they're in the hippocampus. They're also in the cerebral cortex, and not so much there. But if you're talking to lamina 1, that's where the apical dendrites go, part of them. They go up there and they listen to lamina 1 stuff. So they're talking to pyramidal ones, but not the cell body. Granule cell layer. I don't think I've mentioned the term granule cells before, but they're well named because they look like grains of sand. They're little. The cerebellum has lots of them. The dentate gyrus has a lot of them. I used to be able to lecture about the matrix, and people would understand what I was talking about. Not so much now, but they form a matrix. They hold hands. I'm going to show you a PowerPoint I have where I wasn't planning on showing you the granule cells. Oh, what the heck. All right, let's take a quick look at granule cells using Golgi. Remember, I don't like Golgi. I do like Cajal. And these are pyramidal cells. That's a Purkinje cell. These are I have better pictures. But they have globular cell bodies and little arms that reach out, and they literally hold hands with their partners. And they form a matrix. Each of them, they're three-dimensional, so they may have like 8 to 10 little hand-holding. There we go, finally. That's stereotypic. So you have a globular cell body. Those are the granule cells. And out go little dendrites, and there are their hands. And in this picture, you can't quite see them holding hands, but they do. And they don't just, where they hold hands with one adjoining cell, others will join in. My advisor in grad school over coffee one morning, he said, granule cells in the cerebellum are the most numerous cells in the nervous system. Granule cells, cerebellum, which I talk about in relation to Purkinje cells, they are the most numerous cells in the nervous system. And he didn't mention it, but when you look at what they do, they hold hands. And they hold hands in 3 dimensions. And the people they hold hands with other cells. What the heck are they doing? You gotta wonder. I can't answer the question, by the way, but that's important. Okay, so again, the pyramidal cell and the granule cell, they don't get many projections. The pyramidal, though, you don't need to, because you can go to the apical dendrite and get there. So there's figure 6.5, anatomy of the serotonergic system, B cell group. So here's B7, sounds like a vitamin, dorsal raphe, and these 2 bumps here are the superior and inferior colliculus. The cerebral aqueduct is not labeled, but it runs through here. B7 is in the ventral periaqueductal gray. The B8 is the median, and that's not in the periaqueductal gray. It's lower. Why did I make the habenula red? Because the habenula showed up in terms of things modulating I believe, the ventral tegmental area and punishment. And there's the habenula again. I like it because it talks to the periaqueductal gray. It's kind of primitive. It's interesting. It doesn't get the attention I believe it deserves. And it's getting serotonergic input. And just let that wash over you. There's hippocampus, Kapoor, caudate putamen, globus pallidus, amygdala or amygdaloid complex, septal area. Boy, I never would have put that there. Olfactory bulb, neocortex, which you would call the cerebral cortex, particularly lamina 1, cerebellum, geez, everything. And notice the projections to the spinal cord. Raphe magnus is going to be very important for that, and also for descending pain inhibition. And then there's a pretty long discussion. Okay, this has to be dark field again. There's an antibody against the serotonin transporter. And since the transporter has to transport things in the axons show up, and there's dark field. So I had always intended on doing dark field in this chapter. And up here, there's layer 1 of what you would call cortex, neocortex. It has 6 layers. Number 1 definitely gets most of the attention. This white matter, which goes in, eventually a lot of it goes into forming the corpus callosum, not much there. And here we are in the hippocampus, which is very folded up. And look at how dark this area is. There's your pyramidal cells, like nothing. And here's your granule cell layer, nothing. But the rest of the hippocampus gets pretty good innervation. And there's a discussion here of active waking and quiet waking and sleep wave and REM sleep. The book is very interested in sleep. EEG, we've seen that. But of course, these are single cell recordings. And there's orexin again. Orexin keeps coming up in the chapter. Sleep-wake cycles, yin and yang. And that symbol, the yin-yang symbol, is not a bad way of thinking about some of the elements that deal with sleeping and waking. You'll read that. Phasic firing, the dorsal raphe. There's the habendula again. Lots of other neurotransmitters here. Lots of them. So these cells, all the neurotransmitters almost talk to everybody. In the case of the dorsal raphe, serotonergic neurons, many of the major inhibitory inputs use the transmitter GABA, gamma aminobutyric acid. They come from a number of different brain areas. Get ready, including the hypothalamus, substantia nigra, ventral tegmental area, periaqueductal gray, and also cells within the dorsal raphe itself. There's evidence for presynaptic. You have to take a deep breath and remember what that means. You have the presynaptic cell and the postsynaptic cell, and you can have synapses on the ends of axons, presynaptic, glutamate and GABA. Glutamate is a big excitatory amino acid. GABA is a big inhibitory amino acid, and they're playing with each other, and they can affect serotonin intake or serotonin activity in the dorsal raphe. Regulation of serotonin. Where did I go? Oh, I went back. Sorry. No, that was the page I wanted. So this is a good quote. The discussion of neurotransmitter regulation of serotonergic neuron firing has focused on glutamate, acetylcholine, and GABA because of the extensive studies of these particular connections. However, it's important to recognize that there are numerous other neurotransmitters that help regulate serotonergic activity, including dopamine for the ventral tegmental area, norepinephrine from the locus coeruleus, and remember, catecholamines, they seem to have color, substantia nigra, locus coeruleus, and orexin from the hypothalamus. So that's cute. And now it gets nuts. There is a large family of serotonergic receptors, most of which, okay, this is important. Most are metabotropic. And it's very important to remember the difference between metabotropic and ionotropic. And I think I had a question on that. I am just blown away. 14 serotonergic receptor subtypes. I think one of the study questions is list them all. I'm going to make it easier than that on the test because that's insanity. That's like Bob Cratchit with his quill pen and visor doing bookkeeping the night before Christmas. Now, this is important. All of these receptors are metabotropic except for 5-HT3 receptor, which is an excitatory ionotropic receptors. And then there's a discussion of the 1C and how they made a mistake and they fixed it. Okay. 5-HT1As. Now, they reduce cyclic AMP synthesis by inhibiting adenylyl cyclase. Not so amusing to me, but the second obviously is. The second mechanism involves increased opening of potassium channels and membrane hyperpolarization. I'm obsessed with the relative refractor period and why it happens. And it happens because extra ionotropic, not ligand specific, but uh, uh, extra receptors caused by, that are voltage dependent, open, and at rest, at the resting potential, there's a little bit too much potassium in there because the pump is throwing it in and it can't quite get back out. And because there's extra in there, if you opened extra doors for potassium, potassium would leave. And if a positive ion leaves a membrane system, the membrane system gets more negative. So that's what happens during the relative refractor period. And if we have the ability of opening these potassium channels, then at the resting potential, then extra potassium is going to leave and the cell will hyperpolarize. And by hyperpolarizing, its membrane potential will move away from the threshold of activation, making it harder to activate the cell. Therefore, the cell is inhibited. Very important to me. Please try to master that. There's a discussion of drugs that are full or partial agonists for 5-HT1A, and some of these are famous. And there's the most widely used 5-HT1A antagonist is the experimental drug, and you can largely tell it's experimental drug because it uses the letters for the drug company, and there it is. And this should look familiar from an earlier chapter. So there's a metabotropic receptor, and it can affect the potassium channel, opening it, letting potassium exit, which will hyperpolarize the cell. It can also inhibit adenylyl cyclase and have an effect there. And on the right side, that metabotropic receptor can facilitate protein kinase C and very significantly facilitate the release of calcium into the postsynaptic cell, which an earlier chapter mentioned can have second messenger activities. And if calcium cannot enter that cell, if it's an axon terminal, it can't release neurotransmitters. So that's big. So those are both quite big. And now we're getting to 5-H2As, cerebral cortex, striatum, accumbens, and when you think of nucleus accumbens, you should also think of the VTA, ventral tegmental area, and those are areas involved in reward and addiction. And there's lots of drugs, and head twitch. I've seen this one. So giving DOI or other serotonin 2A agonists cause a head twitch. So that's a behavioral indicator of 5-H2A receptor stimulation. More interesting is the fact that DOI and related drugs are hallucinogenic. And this is big in the news now too in terms of potential therapeutic effects. And our federal government was secretly studying this, and allegedly that was going on at UCLA in those rooms, using LSD on volunteers, hopefully. And it's a 5-H2A agonist. Its ability to probably due to stimulation of 5-H2A receptors. Now, again, chapter 15. I think we do get to 15. So we'll see that again. But we're seeing it here, and we're learning about hallucinogens, and we're learning a little bit about the treatment of serotonin. We were obsessed with dopamine D2 receptors for serotonin, for schizophrenia. And now we know that these 5-H2A receptors may be important, and these are more modern drugs. So when I asked you earlier I think I asked you about traditional drugs for the treatment of schizophrenia, these drugs are newer. Clozaril and Risperidol, because they don't just do dopamine receptors, they do 5-HT2As. This has led to the hypothesis that the combination of D2 and 5-HT2A receptor antagonism leads to system improvement in patients with schizophrenia while minimizing side effects, which is a very good thing. Schizophrenics do not like taking traditional antipsychotic drugs. They are no fun. Nobody abuses those. Then there's other receptor types, and migraine, if you suffer from them or know someone who does, this can be extremely debilitating. There are journals. There's a journal named Migraine, and we're still trying to figure this one out. And when you get the right drug, it can be pretty magical. And some of them are serotonin focused. Okay, so now this is a method, which I think was alluded to earlier, iontophoresis. This is transdermal. So you have a device that produces usually a DC current. You have a reference electrode, and you have the stimulant electrode, which is on like the gel pack containing the drug. Obviously, the drug has to have an electric charge, or it will not be driven across the cell membrane. That's pretty magical. It can get things in that other things can't. And there's discussion of 5-HT3 antagonists that are sometimes used for nausea. And that's good. Ooh, I didn't go, and where am I on time here? One second. Oh, geez. Okay. Let me just flip here for a bit, but there's something on my webpage that I wanted to go over. More on knockout mice, like without serotonin, lots of drugs, what they do, largely reviewing what we've already seen. Impulsive behaviors. OCD is sometimes treated with serotonin-related SSRIs. Different kinds of tests, aggression, lots on aggression, types of aggression. Aggression. That's pretty impressive. Impulsive aggression. In a country with lots of handguns, you don't want impulsive aggression. Whereas treatment with serotonergic drugs may be warranted in patients who have been diagnosed with psychiatric disorders that manifest impulsive and irritable aggression, few would argue that we should forcibly medicate everyone who shows signs of violent behavior. I would like them to not be violent. Unexpected death due to epilepsy. There was just a famous case, a Disney actor who apparently suddenly died, and he had epilepsy, and it may be related to this, but I haven't really looked at it. There's fen-phen again, obese patients, anxiety, Buspar. Oh, elevated zero maze. I was going to show you a picture of that. You've seen the elevated plus maze, which looks like a plus, and it has open arms and closed arms. The elevated zero maze is a big circle, like a train track under the Christmas tree, but it's elevated, and part of it is exposed, and there are no walls, and part of it has walls, and anxious animals stay where the walls are. And here we go, pain. This is what I want to close out with, and lots of talking about types and stuff and learning and memory. I'm going to do pain here in a second. I cannot add much. Alzheimer's, boy, I hope we can figure that one out. Serotonin in your gut. We do not, in my professional career, we have not paid enough attention to the gut. And we now are beginning to. and you will profit from that. Trust me. Irritable bowel syndrome, and I think that's it for the chapter. And now we're going to go to something out there. Here are your questions. Some of these are pretty aggressive. I promise I will tone them down. So you go to Tim Cannon's homepage, and there's a sidebar and research-related stuff. And I'm going to show you parts of this talk, Travels of a Functional Neuroanatomist, Pain, Audition, Environmental Enrichment, Sexual Dimorphism. I must have given this talk 20 years ago. Whoops. Yes. So there, I'm sorry, I already had it up. So here we are dealing with pain inhibition. So I've jumped into like slide 50. And by the way, earlier in this, there's a slide that says, "Hello, my pretties." And in there, beautiful pictures of granule cells holding hands. Well, no, not sure about holding hands, but beautiful pictures of granule cells. Okay, so pain inhibition. Opiates, and notice I don't say opioids, are probably the most reliable analgesics available for moderate to severe pain. Sever. Oh my God, a typo. How do opiates work? We didn't know. You would read textbooks and they would basically say opiates mess you up. And when you're messed up, you don't feel pain. Not very useful. But opiate receptors were found. No Nobel Prize has been awarded, and probably won't because it's been a long time now. They're in the brain and the spinal cord, and they were located in areas that were thought to be important for pain inhibition. So they're in the right place to play a role in pain inhibition. There's, you probably can't tell, but look at that for a second. That's a horizontal section. And it's receptor autoradiography, and the hotter, that is, the more receptors there are, the brighter it is. And the label says caudate, but it's really Kapoor. You can see little hotbeds at the base of the olfactory bulbs. Thalamus, of course, thalamus is important for relaying information from the rest of the body to the cerebral cortex, so that's not too surprising. Cerebral cortex has some, particularly though, notice the frontal cortex, prefrontal cortex, a lot of opiate receptors, but we're here to talk about the periaqueductal gray, and it's pretty loaded. The periaqueductal gray, the around the aqueduct gray. Is it opioid? 3 standard tests. Can it be blocked by naloxone or another antagonist? And you'll recall that naloxone, you would know it as Narcan because now it's sold. They used to give it to us, big jars of it for free. It's a receptor antagonist. Does the thing show tolerance, reduced effect with repeated administration? Because that's characteristic of opioids and opiates. And does it show cross tolerance with other opioids and opiates? You don't remember SPA, but SPA is stimulation produced analgesia. I did refer to it in an earlier talk where I put electrodes in the brain and stimulated the brain and inhibited pain. SPA was important for the discovery of opioids in the brain because researchers were saying, oh, geez, they're putting electrodes in the brain. The electrodes are inhibiting pain. That's electricity. There must be a chemical involved, and oh, geez, naloxone blocks it. So there must be endogenous opioids and opioid receptors. Most folks suspected that opiate receptors had not evolved, this is a joke, in the hopes of us running into a poppy plant. And John Libeskind was my mentor. And that picture on the left was, I literally had a candle picture. I was taking everybody's picture with candle. That's John Libeskind. And on the right, that's John's bulletin board. And that guy with the huge head is B.F. Skinner, who gave John his only C. And he kept Skinner's picture on his bulletin board. And Skinner lived at 21 100 North Washington Avenue, but that's not important right now. Dave Mayer is one of his students. They stimulated the PAG. They produced analgesia that was comparable to 50 milligrams per kilogram of morphine. That is a giant dose. It could inhibit the tail flick test, and we saw the tail flick test in a very unreal characterization. That's a spinal reflex. You cut the spinal cord, it would still happen. And if you're stimulating the midbrain and you can inhibit the tail flick, something must be going from the brain to the spinal cord. And here's a discussion: totally spinally, disconnect the brain. So I mentioned all this. Okay, the plot thickens. Huda Keel was John's Libeskind, his student too. She literally took every drug they had on the shelf, and she gave them naloxone, potent opiate antagonist. It could block the analgesia from electricity, stimulation of the PAG. So it can't be blocking electricity. It must be blocking some chemical released by the electricity. So Huda did that. And here's a picture I took. That's fresh tissue. By the way, you can see a little bit of darkening down here. Just imagine it with me. That's the substantia nigra, not very dark in animals. But here's the cerebral ventricle, the cerebral aqueduct. Aqueducts carry water. And a mother's eye, you can see there's sort of a gray area around it. That's the periaqueductal gray. That's in the caudal midbrain, as the book says. And that's where we started stimulating. This is from an earlier textbook. Raphael Magnus shows up in here. Not so important to this talk. So life was simple. The brain had an opioid-mediated descending brain to spinal cord pain inhibitory system. But to believe the simple story, you had to ignore the fact that some labs couldn't replicate the effect of naloxone. They could produce analgesia, but they couldn't block it. I noticed that in Keel's work with some drugs, actually serotonin drugs, she got different results when she was near the dorsal raphe. So I did this. So this is a little bit more rostral. Here's the periaqueductal gray. There's the cerebral aqueducts. There's the dorsal raphe, looking sort of like the picture at the front of the book. This line indicates the division between dorsal area and ventral. We put electrodes straight down the midline. We gave there are only 9 dorsals and 12 ventrals, and we gave them different doses of naloxone. 0.01 times 10, 0.1 times 10, 1 times 10, 10. And you can see that the ventral area, as you gave more naloxone, you had to use more electricity. The threshold went up. So we came to say that the ventral area was opioid mediated, and the dorsal didn't give a whit about naloxone. Whatever that was, it was producing analgesia, it was inhibiting the tail flick, but naloxone didn't touch it. And here's results of a few studies. Dorsal not blocked by naloxone. There's no tolerance. There's no cross tolerance. The ventral blocked by naloxone. There's tolerance. There's cross tolerance. I can't tell you how many times at UCLA I told them, why aren't we concentrating on the dorsal? Because we already know opiates develop tolerance and cross tolerance. We have morphine. What the heck is the dorsal? We didn't know, and I didn't win that argument. How is it connected to the spinal cord? Well, the PAG has almost no connections to the spinal cord, but it does connect to the raphe magnus, nucleus raphe magnus, and the book did mention that. So, hi, my name is Tim. I blow stuff up. So I blew up the raphe magnus with varying degrees of success. Percent destruction of raphe magnus. If you destroyed less than about 50%, and by the way, you may remember the prolactin story with D2 receptors. Not much happens to threshold, but boy, you start destroying more than 50% and to the moon. Analgesia doesn't seem to work. So the opioid form of stimulation-produced analgesia, the ones that comes from the ventral area definitely goes through raphe magnus. But these same lesions did nothing to the dorsal. So that gets the spinal cord. Remember, the tail flick test is a spinal cord test through another pathway. And there's sort of the answer. This is autoradiography again, this time for cannabinoid, more specifically anandamide receptors. And that's it. That's how it gets there. If you block endocannabinoids, you block the dorsal. And that's it. So allow me that little digression. I will not, I'm going to leave the thread open. I might test you on opioid and non-opioid and dorsal and ventral and raphe magnus. I may test you on that, even though it's not at the end of the chapter. And you can, just go back to this and look at it anytime you want. Have a nice day.